Hello, everyone, and welcome to our lecture on C. diff. We're going to be covering it today and also going over some basic bacteriology. First thing we're going to talk about, before we even get into it, C. diff, Clostridium difficile, Clostrioides difficile, all the same thing, just different names and terminology. This is, of course, by the Amherst Medical Center, so make sure to subscribe for more content. So first thing we got to talk about is cells. What exactly are cells? Well, cells are basically the smallest unit of life that makes up everything. We're made of cells. Animals are made of cells, plants are made of cells, and bacteria are made of cells too, except they exist as singular cells. So they're classified as a certain type of cell called prokaryotes. They're single microorganisms. They're unicellular. They're basically tiny little living cells that do their own thing. They act independently. They don't need other cells around them to survive, unlike cells in our body, which do need other cells. Bacteria can be good, they can be bad. They're not necessarily a horrible thing for our body. There's a lot of healthy and good bacteria we have in our body that exist and even help our body to perform certain things and to get certain nutrients in our body. So bacteria have this structure here. This is just a simple version. We can see in the center here, we have where all the DNA of the cell or of the prokaryote is. DNA is obviously needed for all sorts of life. It's what describes what proteins it makes, so basically what the cell does and can produce. So that's called a nucleoid. We also have these little plasmids here. They're basically more DNA, but they're transmissible between prokaryotes. So this means that bacteria have a special function that they can transmit DNA between each other. And this is important for things like antibiotic resistance, because if one bacteria is resistant, it can spread these plasmids to another one that's right beside it. And all of a sudden you have two bacteria that are resistant, and this can just spread all over the place and cause real big issues in a hospital setting and that sort of thing. We also want to see here, we have a couple layers. So basically these three layers, the first one, the closest one here is a cell wall. That's basically just a hard part of the cell that protects against other things. There's also a membrane here. We're going to look at that membrane a little bit closer in a second here. And of course, the capsule is just something that some bacteria have, and it will protect against stuff outside of the environment and make it more, allow it to kind of evade the immune system more than other bacteria. So it'll make it more pathogenic, which basically means it will cause worse disease in the body. So talking about that membrane around the bacteria, there's two types of membranes. There's gram-positive membranes and there's gram-negative membranes. Gram-positive membranes have a thick peptidoglycan layer, and gram-negative cells have a thin peptidoglycan layer. Basically, peptidoglycan is just this polysaccharide or this molecule in the cell that protects it. It acts as like the main molecule that's surrounding the cell, and it mostly prevents stuff from getting in and out and that sort of thing. Provides some structure to the cell as well. Another difference between gram-positive and gram-negative is, of course, there is no outer membrane in gram-positive, but there is an outer membrane in gram-negative, and we're going to see this in an image after the slide. And some applications of this is that gram-positive are generally more successfully treated with penicillins. This is because penicillins target that peptidoglycan layer, and if there's more of it, it breaks more of the membrane up. So the cell will be more likely to, kind of die or not be able to reproduce, not be able to function correctly, rather than gram-negatives where penicillins aren't as successful since there's less peptidoglycan layer. And since removing that peptidoglycan will have less of an effect on the cell. So here we can see the difference. We can see how thick this peptidoglycan layer is in the gram-positive cell walls versus the gram-negative cell walls, which just has a thin little layer here. And in addition to the first membrane here, it has a second outer membrane here. It also has LPS, but that's something we'll be talking about another day. So endospores, one of the last things we're going to talk about for bacteria here. Endospores are specialized sort of bacteria. They're produced by bacteria that are like active and stuff, but they're a special one that can remain dormant, and they stay in like really harsh environments and can survive all sorts of conditions: hot, cold, dry, wet, all sorts of things. But what allows them to do this is they have a really hard peptidoglycan coat around them that makes them resistant to not only these environments, but also really common antimicrobials like all alcohol sanitizers and such. So what these things will do is they'll stay in environments until they get activated by warm temperatures, moist temperatures, whatever its ideal conditions are. So it can be it can stay on like door handles or stuff as this form until somebody might like touch their hand or touch the door handle and touch their hand to their mouth. And then once it gets into their mouth, it could activate and then become an active bacteria that can cause disease. Because endospores themselves, since they're dormant, they're not going to cause disease. But once they activate and become active germinating bacteria, that's when they can cause disease. So these endospores can be inactivated with soap and water because that'll cause friction and stuff, and that will just cause the membranes to burst no matter how hard that coat is. So hand washing is always great. Also, some extreme pHs like bleach can also get rid of it, in addition to some extreme temperatures. So like if you put it in an autoclave with some pressure, autoclave is basically how you clean things in a hospital properly. If you put it in there, it should, if the autoclave is working properly, it should be inactivated. And it's important to say inactivated rather than dead, because the endospore really isn't living in the first place. It's only active or inactive. So finally onto the topic of Clostridium difficile after all that intro. So Clostridium difficile is a gram-positive, meaning it has that thick peptidoglycan layer. It also produces those endospores that can survive in all sorts of environments. It is transmitted via the fecal-oral route, meaning that if you have anything that gets into your mouth, any of those spores or the bacteria, it can infect you that way. And that fecal matter can contaminate all sorts of things with spores or active bacteria. These spores, endospores, can vary how long they can survive for, but generally it's like years, and that's true for Clostridium difficile. So like 3 years later, after a patient had C. diff and like they got some endospores on a door handle or something, or the floor, if it hasn't been properly cleaned with bleaches or something that was going to kill those endospores can still activate. They can still activate if they're exposed to the proper environmental conditions. So spores, when they get into the body, they germinate in the small intestine, and this is where they're going to cause disease from. The activated bacteria will create exotoxins, and exotoxins are basically these specialized particles or molecules that the prokaryotic cells will produce. And these exotoxins are going to damage the intestinal mucosa, so they're going to damage the intestine and cause inflammation and that sort of thing. That is how they're going to cause disease to the individual. And these are also, Clostridium difficile is also a really important nosocomial infection, meaning it spreads in health facilities, so like hospitals, all that sort of thing, nursing homes. Yeah. So risk factors for C. diff. Consuming uncooked meats and produce can get you this, because in soil there's a significant amount of C. diff spores. So if you don't properly wash your lettuce or something, or some meat gets contaminated, you can get Clostridium difficile spores introduced into your body that are not spores, endospores introduced into your body that way. Hospitalization is also a risk factor, because in hospitals you're exposed to all sorts of diseases, because that's where people who are sick go, and they're going to be generating these endospores, and these endospores, even though we try really hard to follow asepsis and all sorts of stuff, sometimes this stuff still gets around and can't clean everything. So there's more presence of these endospores and all sorts of other things in hospitals. So that's a risk as well. Recent antibiotics or antibiotic therapy. This can cause the normal flora of the intestines to be unbalanced or damaged. And when the normal flora is damaged, this presents an opportunity for bacteria like C. diff that can cause bad things, can cause infection and disease to grow, since there's less competition from these normal flora. And normal flora, or natural flora, is basically the good bacteria, the happy bacteria, the bacteria that's not going to kill you and that's helping you or at least not hurting you in your daily life. But something to note is that this bacterium, so C. diff, can exist asymptomatically in the intestines. It's in probably, I think it's around 5 or 10% of the population. So it's not causing disease in these people since it's asymptomatic, but it still exists. It'll obviously not in large numbers, otherwise the exotoxin would be causing some damage, but it might exist in like a small part or in a small population in your intestines. So when your normal flora might be disrupted, this small little amount of bacteria might grow to a big amount of bacteria if the normal flora is damaged enough that it presents an opportunity for this bacteria to expand and exponentially grow. So that's when the bacteria might become pathogenic after the normal flora is disrupted by something like antibiotics. So some early symptoms of a C. diff infection would be diarrhea, and it would be watery. You might also have fever, loss of appetite, abdominal pain, or nausea. All basically signs that your intestines are getting inflamed and damaged by this exotoxin this bacteria is producing. So we're going to be looking at some colonoscopy pictures quickly. So this is just to set the baseline here. This is what a healthy, non-infected, happy colon's looking like. It's nice and pink. So no inflammation, no discoloration, happy colon. And this is what happens if you get late or severe stage C. diff infections. On the left here, we have PMC or pseudomembranous colitis. Basically, you can see all this inflamed and damaged intestinal tissue here. It's all yellow. There's pus buildup. It's all inflamed. This means that there's bacteria growing all over here, and the immune system's trying to fight it. Lots of exotoxin being produced. This obviously going to be really painful. You're going to have lots of symptoms. And if this isn't treated, it might develop to toxic megacolon. And toxic megacolon is basically when this gets even worse. This is basically the worst thing you can get from C. diff besides death, because this can cause, this can cause death. It's basically the gangrene of the intestinal tissues. And once tissue becomes gangrenous, it has to be surgically removed. There's no bringing back this dead tissue once it dies like that. And dead tissue is obviously going to cause other issues. It's going to cause inflammatory things in your blood to really spike up. It's going to cause things like sepsis if you leave it too long. Very bad in a lot of ways. And you need immediate intervention for this. And this too, of course, to prevent toxic megacolon from developing. And just to go back quickly, you can see how this tissue has become black, and that when tissue becomes black, that basically means it's dead and gangrene has happened. Basically how you would determine the difference between these two things and additional swelling as well. So treatment for C. diff infections like this, conventionally you would want antibiotics. So vancomycin is a specific type of antibiotic we might use for this. I'm not expecting people to memorize the antibiotics that we'll be going over that later on, hopefully. As well, you want to be ending unnecessary antibiotics. So like if you're taking a penicillin for whatever reason, that might be affecting your colon flora. So we want to be cutting that off unless it's necessary for an infection that's still persisting, because these are going to be damaging your normal flora and making the bacteria have more opportunity to grow because there's less other bacteria competing with it. Those are the conventional methods, and some non-conventional methods. These are best for chronic infections especially. Would be probiotics. What probiotics do is they promote your normal flora to grow. You see them in yogurt and stuff. Basically things that are going to help the bacteria in your gut be happy. All the healthy bacteria is going to be growing. The best result, though, or the most successful treatment, especially for chronic infections, but it's also applicable to acute infections as well, is fecal implants. And what fecal implants are is basically this wonderful flora that you eat. You can think about it that way, but it's basically somebody else's fecal matter because it has all the wonderful bacteria in it. And basically the point of it is to reestablish healthy bacteria in your gut again, because for whatever reason your normal flora isn't growing and C. diff's growing all over the place. You're going to try to reintroduce that healthy bacteria back into your gut, and hope that it outcompetes that C. diff and reestablishes healthy normal flora and subsides the infection. So on to our summary now. We want to remember that Clostridium difficile, or Clostridium difficile, or C. diff, whatever you want to call it, is a common and harmful bacterium that is found in many environments due to its production of endospores, which are those things that will stay in any environment almost and are really hard to kill. We also remember that antibiotics and fecal oral exposure are the biggest risk factors for getting the bacterium. Fecal oral exposure, especially if you're in an environment that may contain those endospores, like a hospital or in soil. We want to remember that severe disease can lead to gangrene or toxic megacolon, or gangrene of the colon. And finally, we want to remember the treatment for this, which should include fecal implants to try to establish that normal flora. We want to be thinking about that normal flora and making sure that it gets back and becomes healthy. And yeah, that is it. If you want more info, you can check these sources. I also use this source a lot in the research and everything, so I definitely suggest it if you want to be looking for more. Thanks for listening, you guys. Make sure to subscribe and like, and hopefully we'll be making another one of these videos soon. Bye!